Hello, I'm Nicholas Fernandez. I'm a computational scientist at the Human Immune Monitoring Center at Icahn School of Medicine at Mount Sinai. And this video tutorial is going to walk you through a short notebook visualizing some CITE-seq data that is made publicly available by 10x Genomics. So we're going to start off with the Clustergrammer2 notebook GitHub repository. And if we scroll down, you'll see the README shows you several examples we have preloaded. But for our example, we're going to click the Launch Jupyter Lab badge to launch a Jupyter Lab instance on a service called MyBinder. And in the interest of time, we've preloaded the notebook and Jupyter Lab here. So if you look at the URL, you'll notice that it's being hosted on the cloud by MyBinder. And we've gone ahead and navigated to the notebook 4.1, 10K PBMC CITE-seq, visualize ADT and GX data. So this notebook is going to use Clustergrammer2 to visualize this CITE-seq dataset. So this dataset's pretty interesting in the sense that it actually allows you to measure two distinct properties of single cells. So not only like our previous example, the 3.0, 2700 PBMC example, you're not only measuring across the entire genome, the expression of genes in single cells, but you're able to measure surface marker proteins using a process that's called, we refer to as CITE-seq, and in the context of 10x, it's called feature barcoding. And so in this case, you're getting like two distinct measurements from your single cells. And this notebook is going to allow us to view our single cell data in these two different spaces, the protein space and the gene expression space. So in our notebook, we start off by loading Clustergrammer2, some helper functions, and then we proceed to load our data. So the previous notebook 4.0 actually does some pre-processing and does some clustering of the cells, but we're going to be viewing the outputs here on MyBinder. So we start off by loading our data. We're loading it from a file format called Parquet, just as a demonstration of how we can use this general purpose columnar format for our data set. And we load the protein data, which we're referring to as ADT, and the gene expression data here. So we can see the size of our data sets. So our protein data has 14 rows and 7,800 columns. So this corresponds to the 14 proteins that were measured and the almost 8,000 single cells that were measured in this data set. And our gene expression data, we're loading a small version of it where we're only including the top 100 variable genes across our cells. So we have our data set is, for gene expression is 100 by 7,800. So we're just normalizing our gene expression data here to enforce a common UMI level across all cells. And then we're going to take two views of our data. So the two heat maps that we're generating with Clustergrammer are a view in the protein space and the gene expression space. And what's nice about Lab is that you can actually right-click a cell and tell it to create a new view of the output, which we've already done. And so I'm going to scroll up here to ADT. I'm going to close our sidebar section here, and then I'm going to drag this tab that we've already opened up here. And so now it's just a little bit more convenient to view these two things horizontally next to each other. So here on the left, we have our ADT data, and on the right, we have our gene expression data. And the cells are labeled using the same categories. So in the previous notebook, we went ahead and clustered our cells based on, minimize that, okay. So in our previous notebook, we clustered our cells based on surface marker protein, and we identified 7 distinct clusters here, so 1, 2, 3, 4, 5, 6, 7. And we've labeled them based on their protein level. So for instance, you can see that this cluster of cells is high for CD8A. We went ahead and labeled as CD8 T cells. And CD14 expressing cells, we just manually labeled as myeloid CD14. So we have these 7 different clusters of cells in the protein space and in this 14-dimensional space. So we can go and investigate here and reorder based on a particular marker. And then we can see that which cell types are highly expressing a particular marker. So we can see different patterns in our data. We can reorder our columns, our cells based on overall expression to sort of see if they're, or here we put in cluster view, but we can rank by expression to see, like, are there some cells that are hardly expressing any proteins? These might maybe not actually be cells, they might just be debris. And are there cells that are expressing a lot of proteins, more than you would expect? These over here on this edge might actually be doublets that haven't been removed. And Clustergrammer just allows you to really intuitively explore this data. But what's nice about this view is it really emphasizes the fact that we are getting these two parallel information sets. So on the right here, we're looking at the top 100 variable genes across our 7,800 cells, and we have the same coloring scheme. So for the most part, the cell type labels that were defined based on protein are preserved in gene expression space. So if we ever hover over here, we see we have our myeloid cells over here. In this kind of orangish, we have NK cells. And so there's a lot of structure that's preserved. But if you look at the dendrogram, you see it's not completely preserved. So there's a mixing of different cell types together. So this can tell us, it can help us identify cell types or cell states, for instance, that are not captured at the protein level, that are captured in higher dimensional gene expression space, or it might point to certain advantages or disadvantages you have in the two different measurements. So some you might want to define your cell type based on surface marker and then transfer that to gene expression, or you might want to do the opposite, define your cell type based on gene expression and see how well it agrees with surface marker space. In this case, we went from protein to gene expression, but you could do both. And then we can just go in and explore the specific differentially expressed genes in these clusters and start to understand the relationship between our data. And because these are exactly the same cells, we can just reorder by cell type in both cases, and we're going to get exactly the same ordering, and we can see how well or not well resolved our data is in these different data sets. So we encourage you to explore the notebook, try different parameters on the pre-processing version of it, and we hope you enjoy. Thanks.